Welcome to BioBits and Bytes.
And welcome to another deep dive.
Today we're plunging into a topic that really rocked the scientific world just a few years back. It's fundamentally changing how we understand the machinery of life itself. We're talking about proteins and their shapes.
That's right.
Have you ever wondered why unlocking the precise, intricate 3D form of these tiny cellular machines, proteins, is so absolutely central to understanding biology?
It's a great question. You can think of proteins not just as like building blocks, but more like tiny, complex pieces of biological origami, or maybe incredibly specialized Lego structures.
Legos, I like that analogy.
Yeah, and each protein needs to fold into this very unique, specific 3D shape because, well, that shape is its function. It determines how it binds to other molecules, how it speeds up reactions, builds things in the cell.
If the shape's wrong.
It just can't do its job. Simple as that.
Exactly. And for decades, maybe, half a century, figuring out that exact 3D shape just from the linear string of amino acids, basically translating that 1D sequence into a 3D structure that's been one of biology's toughest, most persistent challenges.
Oh, absolutely. The protein folding problem, they called it.
And computationally predicting it felt like, well, a very distant dream for a long time.
It really did. Progress was hard-won, often slow. It seemed to happen in fits and starts,
Which is precisely why the whole scientific community was just sent into a state of shock, really, back in November 2020.
That was the CASP moment.
Exactly. Google DeepMind's AlphaFold 2 enters the CASP competition. That's the critical assessment of structure prediction, the field's big proving ground. And they didn't just win. They absolutely dominated. Unprecedented accuracy.
The results were just, people use words like astounding, a miracle. The consensus was immediate. They had, for the most part, largely solved the protein folding problem.
A problem that seemed decades away from being solved just moments before.
It really felt like that. A huge barrier just suddenly crumbled. As John Molt, one of the CASP founders, famously put it, this is not an end, but a beginning.
And that's our mission for this deep dive. We want to unpack AlphaFold 2. Understand the core ideas, how it worked, explore why its success was, frankly, so surprising to so many people.
And look at its impact. It's been revolutionary for biology and especially for drug discovery, which is obviously critical.
Right. And we'll bring you right up to date with the latest developments, too, including the arrival of its successor, AlphaFold 3.
We've gathered quite a bit of material for this, reports from the CASP competitions, some technical deep dives into AlphaFold 2's architecture, analyses of its real-world impact, and, of course, the recent news and discussions swirling around AlphaFold 3.
Our goal here is really to guide you through all this.
Yeah.
We want to distill the key insights so you can quickly get a solid handle on this pivotal moment in science, even if you don't have a deep background in, say, biology or AI.
We'll try to break it down.
We're going deep, but hopefully we'll make it understandable.
Let's get into it.
All right, so let's start right at the foundation. Why do protein structures matter so much? We said they're machines, but what does that really mean in practice?
Well, they're the functional workhorses, right? They carry out almost every critical process in our cells. I mean, think about anything happening inside you right now, digesting food, building tissues, sending signals.
Moving muscles, fighting off infections.
All of that. It's proteins doing the heavy lifting. Enzymes that speed up reactions are proteins. Transporters moving things across membranes are proteins. Antibodies defending you, proteins. Receptors receiving signals, proteins.
So their shape is everything.
It's everything. That specific 3D shape is what lets them interact with other molecules in a super precise way, like a key fitting into a very, very particular lock.
Okay, so that sequence structure-function relationship is absolutely fundamental. You have the DNA sequence. That determines the linear chain of amino acids.
Which we can get relatively easily now, thanks to genomics.
Right. And that sequence then folds up into a unique 3D structure.
And that structure dictates what the protein actually does, its function.
But while getting the sequence got fast and cheap, figuring out the 3D structure experimentally, that remained the bottleneck, didn't it?
Oh, massively. Methods like X-ray crystallography, NMR, cryo-EM, they're powerful, no doubt. But they are technically really demanding, incredibly time-consuming, and honestly, they often just don't work for every single protein out there.
I remember reading it took pioneers decades, literally decades, to solve the first few structures like hemoglobin or myoglobin back in the mid-20th century.
And those were Nobel Prize-winning achievements. It just highlights how incredibly challenging the experimental side was and still can be.
It really shows the scale of it. And yet, even back then in the 50s, there was this crucial insight from Christian Anfinsen.
Ah, yes, Anfinsen's dogma, a cornerstone.
What was the core idea there? Can you remind us?
His work was groundbreaking. He showed that a protein, even if you unfold it, denature it, it could spontaneously refold itself back into its correct native 3D structure. And it did this based only on its amino acid sequence and the surrounding chemical environment.
So the blueprint for the 3D shape
Was entirely contained within that 1D sequence. All the information needed was right there.
which logically makes you think the prediction problem should be solvable computationally. If the sequence has the blueprint, computers ought to be able to read it right.
That was absolutely the hope. And Infansen's work essentially split the big protein folding problem into three main questions. First, the one we're mostly focused on today. Can we actually predict the structure just from the sequence?
Computational challenge.
Right. Second, what is the underlying folding code? You know, the actual rules or physical principles that govern how that sequence dictates the final shape.
A third.
What's the actual folding mechanism, like the step-by-step physical journey a protein takes to get to its final folded state in a real cell?
Okay, and those early computational efforts starting way back in the 60s, they tried to tackle that prediction question.
They did, but they were really hampered. Limited computing power back then, and frankly, a less complete understanding of all the intricate forces involved in folding.
I can only imagine trying to simulate thousands of atoms interacting from scratch back then.
Yeah.
And the source we looked at mentioned this kind of tension, this contrast between the super slow, meticulous experimental work and maybe some faster, possibly overly optimistic claims coming from the computational side early on.
Yeah, there was definitely some, let's say, healthy skepticism or necessary friction, maybe. The experimentalists knew just how incredibly hard this was in the lab, and the computational folks were exploring new territory.
And this friction, this need for objective evaluation, is what led to CASP.
Exactly. The critical assessment of structure prediction, co-founded by John Malt and Krzysztof Fidelis in the early 1990s, precisely to address this.
To cut through the hype and provide a real test.
Precisely. It set up these blind tests. Groups worldwide would predict structures for proteins whose experimental structures were known but not yet public. CASP basically forced computational methods to face experimental reality head-on, an unbiased assessment of accuracy.
No hiding.
None. The source paints a pretty vivid picture of the CASP conferences at Asilomar, presenters showing their results, and apparently attendees would literally stamp their feet if they thought a method wasn't being presented honestly.
Wow, intense.
But it brought much-needed discipline. It really pushed the field forward.
Sounds like it. But those early CASP rounds, the prediction accuracy wasn't exactly setting the world on fire, was it?
Huh.
The source quotes Molt saying about some early results, "Random is a good word."
Slight chuckle. Yeah, the early days were definitely humbling, but, progress was made, slowly, steadily. And one technique that emerged fairly early and proved pretty useful when it applied was homology modeling.
Homology modeling.
Yeah.
Right. That's predicting a structure using a known structure from a related protein as a kind of guide or template.
Exactly that. The basic idea is if two proteins have similar amino acid sequences, they probably fold into similar 3D shapes. So you take your target sequence, search databases for known structures with similar sequences.
Find a match, a template.
And you use that template structure to build a model for your target protein. The source gave that nice example with myoglobin from different animals: human, elephant, tuna, pigeon.
Right, the similarity varied.
Yeah, human and elephant myoglobin are like 80% identical. You get a great model. Tuna at 45%, pigeon at 25%. The models get less accurate, but you can often still get a useful structural idea. And historically, CASP targets were often categorized based on whether these kinds of templates were available.
Okay, so homology modeling works well when you have a close relative with a solid structure.
Mhm.
Makes sense. But what about the really hard cases? Proteins that are totally novel, no known structural relatives, no templates.
Yes, that was the much tougher nut to crack,
Mm.
often called de novo prediction or template-free modeling. For many years, progress there was significantly slower, until a really key breakthrough started gaining traction in the 2010s. It involved leveraging sequence information in a much more powerful way, looking at coevolution.
Coevolution. Okay, that sounds like it pulls from evolutionary biology.
Mhm.
How does looking at evolution help predict a 3D structure?
It's a really elegant idea, actually. You start with what's called a multiple sequence alignment, or MSA. That's essentially a big collection, a stack, of many sequences for the same protein, but taken from lots of different species, maybe dozens, hundreds, even thousands.
So you see how the protein has changed over evolutionary time.
Exactly. And as these proteins evolved, mutations happened. But here's the key. If two amino acids in the sequence need to be close together in the final 3D structure to keep the protein working properly, maybe they form a bond, or just need to pack together tightly, then they are under evolutionary pressure together.
Okay, so if one of them mutates in some species.
The other one might also have to mutate in that same lineage to compensate, to maintain that crucial interaction. Maybe a positive charge mutates, so the negative charge it was paired with also has to change to maintain the balance. You see this pattern of correlated mutations.
I get it. So even if amino acids A and B are really far apart in the 1D sequence, if you consistently see them changing together across many, many species in your MSA.
It's a very strong signal that they are probably physically close, interacting in the folded 3D structure. Evolution has basically left clues about the contact map for you.
That's incredibly clever, using evolutionary history to find structural contacts. And the source mentioned this was a huge leap forward.
It really was. People developed sophisticated statistical tools to pull these correlated mutation signals out of the MSAs. And the accuracy of predicting which amino acids are in contact jumped dramatically. From maybe 20-24% accuracy in the early 2010s, it went up to 47% by 2016, and then hit around 70% by 2018.
Wow, 70% contact accuracy. That must have made a huge difference for building models, especially those template-free ones.
Absolutely. Methods incorporating this co-evolutionary information performed extremely well in CASP13 back in 2018. It was a major step.
Okay, so by 2018, the field had pretty sophisticated tools. Homology modeling when possible, powerful coevolution analysis for harder targets. Progress was definitely being made, building on all that prior work. It felt like, as the source put it, maybe punctuated equilibrium, periods of slower progress than these exciting bursts like coevolution.
That's a really good description of the state of play then, and it perfectly sets the stage for the next punctuation mark, the unprecedented one, CASP14, announced November 2020.
The CASP14 shockwave. I definitely remember the buzz around that. What was the immediate reaction like within the scientific community?
The source uses words like shock, even denial, and that feels accurate. AlphaFold 2's performance was just so far beyond what anyone was expecting, even given the progress in CASP13.
Better than the coevolution methods.
Dramatically better. They were achieving accuracy levels that were comparable to, and in some cases maybe even better than, experimental methods for quite a few targets. It just felt like this massive problem, the protein folding problem, which had been largely unsolved for 50 years suddenly, was largely solved pretty much. When John Molt announced that AlphaFold 2 had largely solved the protein folding problem, there was apparently this stunned silence in the room, then just immense excitement. It was genuinely a field-defining moment.
Breathtaking.
Yeah.
And the person leading this effort at DeepMind was John Jumper. Tell us a bit about him. His background seems interesting.
Yeah, Jumper's path wasn't entirely conventional for leading a biology breakthrough. He started in physics and math, interested in fundamental questions, but he apparently found the pace of some experimental physics a bit slow for his liking. Then he worked for a time at D.E. Shaw Research. It's a quantitative hedge fund, but they have this serious computational biology division, DSURS. They use custom-built supercomputers for incredibly long, detailed protein simulations. So he got deep exposure to proteins and massive scale computation there.
So physics, computation, proteins, quite a mix.
Exactly. Then he went back to grad school, specifically wanting to use AI and machine learning to accelerate the kind of simulations he'd done at D.E. Shaw, but, without needing their custom supercomputers. He saw the potential for deep learning.
And that path led him to DeepMind.
And eventually to leading the AlphaFold 2 team.
All right, let's dive into how it actually worked, at least the high-level intuition. The source gave a nice bird's-eye overview, which sounds perfect.
Right. So at the very start, like pretty much all prediction methods, AlphaFold 2 takes the protein's amino acid sequence as its input.
The basic 1D string, standard stuff.
Then it does an initial processing step. Very standard bioinformatics pipeline stuff, actually. It searches those huge protein databases to build that multiple sequence alignment, the MSA we talked about, finding all the evolutionary cousins.
Using the coevolution idea later.
Exactly. And it also searches for any known protein structures that might be similar, potential templates for homology modeling. So these initial steps gather a ton of information about the sequence itself, its relatives, any structural clues from known proteins. And this generates an initial sort of pair representation, a first guess about which amino acids are likely to be near each other in the final structure.
Okay, so it's still using those fundamental tools, MSAs, templates that the field had been developing for years.
Absolutely. It builds on that foundation. But then it feeds all this information into the core of the system, the evoformer module. And this is where, the deep learning magic really starts to happen.
The evoformer. Okay.
Yeah.
What's the key idea? What makes it different?
The really central, powerful idea in the evoformer is how information flows. It's not just a one-way street. Information flows back and forth iteratively between the representation of the sequence information, the MSA data, and the representation of the structure, the geometry, the evolving model of contacts and positions.
Like back and forth. How does that work? I thought previous models mostly took sequence info and just predicted a structure as the output.
That was generally the case. But here, in the evoformer, that intermediate structural hypothesis, the pair representation, predicting contacts, isn't just an output. It's also fed back in to help the model refine its understanding of the sequence data, the MSA. And then that refined understanding of the sequence data helps improve the structural hypothesis. And that improved structure feeds back again to help interpret the sequence. It goes round and round through many cycles. The published model had, I think, 48 of these blocks or layers.
Okay, let's pause and simplify that part again. That iterative refinement sounds crucial. Can you give us a sort of analogy?
Yeah, let's try. Think back to the coevolution idea again. The model looks at the MSA, spots a correlation between amino acids, say, A and B. That suggests A and B are close in 3D. Okay, that's an initial structural hypothesis.
Step one.
Right. Now, the evoformer uses that hypothesis. It says, okay, if A and B are close, maybe amino acids C and D, which seem to be near A and B in my current structural guess, maybe they're related in some way too, even if it wasn't obvious just looking at the raw MSA initially.
So the structure informs the sequence analysis.
Exactly. The model takes the structural context back to the MSA data and looks, again, maybe for more subtle correlations involving C and D that it missed the first time. If it finds new evidence for CD correlation in the MSA, maybe that strengthens the hypothesis that C and D are close. That, in turn, refines the structural model, bringing C and D closer.
And that refined structure then helps it look at the MSA again.
And again and again. It's this constant dialogue, this conversation between the sequence information and the geometric information, each perspective informing and improving the other iteratively.
I think I get it now. It's like it's building the map, the structure, and simultaneously using the map it's building to better read the terrain data sequences. That constant feedback loop is the key innovation.
That's a really good way to put it. That iterative refinement is absolutely central. And this whole evoformer module, architecturally, it's based on a type of deep learning network called a transformer.
Transformers, yeah, they became huge after that. Attention is all you need. Paper, right. What's the main idea there? Attention. How does that apply to folding proteins?
Right. Attention is the core mechanism. Simply put, it means the network learns to weigh the importance of different parts of its input data when making a prediction. Instead of treating everything equally, it learns to pay attention to the most relevant bits.
Like, if you're translating a sentence, the network learns which words in the source language are most important for predicting a specific word in the target language.
Exactly that kind of idea. Or in image captioning, focusing on the pixels representing the dog or the frisbee. For protein folding, the attention mechanism helps the network figure out which pairs of amino acids are most informative, which patterns in the MSA columns are critical, or which parts of a template structure are most relevant for predicting the position of a specific amino acid. It helps it zero in on those crucial relationships, like the correlated mutations, even if they're between amino acids way far apart in the sequence.
So attention helps it cut through the noise and focus on the critical long-range interactions that define the 3D structure.
Precisely. Transformers are powerful because they can process the input data all at once, not step by step like older networks. This lets them capture these long-range dependencies really effectively, which is vital for proteins. Plus, they tend to train faster and more stably.
But didn't the source mention a downside? Something about computational cost?
Yes, the original transformer design has a computational cost, particularly memory, that scales quadratically with the length of the input sequence. For very long protein sequences, or more importantly, very deep MSAs with thousands of sequences, this gets incredibly expensive.
I mean, serious hardware.
Absolutely. Like Google's specialized TPUs or banks of powerful GPUs, DeepMind used clever architectural tricks in the evoformer, like factorized attention, calculating attention in smaller chunks, row by row and column by column for the MSA to help manage this cost. But it still required enormous computational resources to train and run.
Okay, so the evoformer is basically these two interconnected transformer-like networks, one looking at the MSA, one at the pairwise interactions, constantly talking to each other through maybe 48 cycles, using attention to guide the process, and iteratively refining both the sequence understanding and the structural prediction.
That's the core intuition, yes. And then, after the evoformer has done its work, there's a final structure module that takes these highly refined internal representations and translates them into the actual 3D coordinates of all the atoms in the protein. But yeah, the evoformer is where the really novel computational heavy lifting happens.
Got it. Okay, that gives us a sense of the architecture. But when AlphaFold 2 first burst onto the scene, there was all this buzz, right? People were wondering, did DeepMind discover some, deep secret principle of protein folding, some hidden law of physics or biology that everyone else had missed?
That was definitely the big question floating around. Had they found some fundamentally new piece of the folding puzzle?
But the source we read suggests the secret sauce wasn't necessarily a brand new biological theory, but something else.
Yeah, that's the interesting conclusion the author came to after really digging into the published paper and all the supplementary details, and it's a view shared by many in the field now. The consensus seems to be that the real secret sauce was less about a single undiscovered principle and more about DeepMind's superb engineering.
Engineering,
Yeah.
not some magical biological insight.
Primarily engineering, it seems. Now, the architecture itself was brilliant and novel, no question, but the underlying ideas it was built on, using MSAs, using templates when available, leveraging coevolutionary signals, the concept of iterative refinement, these were ideas that had been percolating and developing within the wider scientific community for years.
So other groups were thinking along similar lines.
Many groups were exploring related concepts. The breakthrough wasn't necessarily finding a single hidden key, but DeepMind's incredible ability to take these existing and emerging ideas, combine them in a novel way, and then implement them at an unprecedented scale with extreme efficiency and robustness. It came down to their access to compute resources and their top-notch know-how in deep learning engineering.
So it's about the execution. Building this incredibly complex system, training it effectively on massive data sets, that was the differentiator.
It seems so. An architecture like AlphaFold 2, involving, as the source says, innumerable combinations of matrix products and countless subtle design choices. You don't just dream that up and have it work perfectly. It requires intense engineering effort, many cycles of trial and error, constant tuning, optimization.
And that kind of large-scale experimentation, that requires huge resources.
Huge resources, both computational power and human expertise. The source makes a really strong point here. An academic lab, even one with very similar theoretical ideas, simply would not have had the resources and engineering know-how to implement them in practice at the level DeepMind achieved.
Because DeepMind had Google's backing.
Exactly. Financial backing, access to cutting-edge hardware like TPUs, and critically, a large, incredibly skilled interdisciplinary team. Statisticians, biologists, chemists, software engineers, AI researchers, all working together very closely. One person from the team was quoted saying something like, "Things that would have taken me months to do as a PhD student, I was doing in a single day at DeepMind."
That difference in speed and scale is just staggering. So it underscores that breakthroughs like this aren't always just about the cleverest initial idea anymore. It's often about the resources and expertise to actually build the incredibly complex thing that realizes the idea. What do you think about that? Does it change how we view scientific progress?
I think it's a really crucial point for science today. Individual brilliance is still vital, of course, but pushing the frontiers, especially in fields that rely heavily on computation and massive data, increasingly demands these huge collaborative efforts, significant funding, and world-class engineering alongside the core scientific insights. It doesn't diminish the achievement at all, but it does highlight that scale and engineering muscle, often built on community foundations, can be major drivers of breakthroughs now.
It's a really profound shift. Okay, so DeepMind builds this incredibly powerful, resource-intensive tool.
Yeah.
What happened next? They had the code. What did they decide to do with it?
Well, they made what many consider a landmark decision. They open-sourced AlphaFold 2. The source gives them significant credit for this, saying They decided to do the right thing, and the full code became freely available on GitHub for anyone to access.
Which must have been huge news for researchers everywhere, making this revolutionary tool accessible. But
Yeah.
accessible how? Could I just, download it onto my laptop and start predicting protein structures?
Just chuckles slightly. Yeah, not quite that simple, unfortunately. As the source points out, it's definitely not like installing a typical app. It needs serious computational muscle.
Okay.
You need a pretty powerful server or access to cloud computing resources. Just downloading the necessary databases, the sequence libraries, the structure templates takes up about 2.5 terabytes of disk space.
2.5 terabytes. Wow.
Yeah, and trying to run it on a standard laptop, especially one without a beefy GPU with lots of memory, is likely just going to crash or run incredibly slowly.
I bet a lot of excited scientists discovered that the hard way.
I imagine so. However, the source clarifies that if you do have access to, say, a research workstation with a high-end GPU, something like an NVIDIA Quadro RTX 6000 was mentioned, or a server with a lot of RAM, maybe 300 gigabytes or so, then running predictions, even for reasonably large proteins, becomes quite feasible.
So usable for labs with decent infrastructure.
Yes, and DeepMind did put significant effort into making it easier to use. They provided things like Docker images and scripts, which help package up the software and its dependencies. This means it can be deployed more easily on institutional high-performance computing clusters, using systems like Singularity. So while it's not trivial, it is genuinely usable by many research labs and institutions around the world.
Okay, that's good to know. So the code is out there, researchers are using it. What's been the impact? Let's talk about academic research first. The source made a really provocative comment suggesting AlphaFold 2 may become an indictment of academia. That sounds pretty strong.
It is a strong statement, yeah. And it touches on some really complex and sensitive issues within the academic system. The source points to some widely acknowledged challenges in academia. Things like intense pressure, sometimes unhealthy work cultures, issues around funding and stability, precarious postdoc positions, concerns about work-life balance, job security.
Problems many academics are familiar with.
Right. And the argument the source makes is that, in parallel, industry, particularly the big R&D labs at major tech companies, the FAANGs and others, isn't just doing applied work anymore. They're doing arguably some of the best basic research in fields like computational biology, and often they're doing it with better working conditions, better pay, and crucially, access to the kind of massive resources that made AlphaFold 2 possible in the first place.
So the worry is a brain drain. How do you persuade talented young researchers to stick with a tough academic path, with its funding struggles and uncertainties, when industry offers potentially better conditions and the tools needed for the most cutting-edge projects?
That's exactly the dilemma posed. How do you convince someone to navigate years of temporary contracts and grant writing stress in academia when an industrial lab might offer stability, higher salary, and access to the kind of infrastructure needed for something like AlphaFold? There's a concern that industry could become the primary place for all such ambitious research, pulling talent away from the more exploratory, fundamental questions traditionally tackled in universities.
And academia has historically been where many entirely new research problems were defined, right? Frameworks like CASP itself originated there. What happens if the people best equipped to define the next big problems are increasingly drawn to industry, knowing industry might be better positioned to solve them quickly anyway?
It's a really tough conundrum. The source emphasizes it's not about pitting industry against academia. Ideally, they should complement each other. But it does argue that academia needs to seriously look inward and address some of its systemic issues if it wants to remain a competitive and attractive environment for conducting high-risk, high-reward basic research.
A really critical discussion for the future of science.
Well,
Okay, stepping back from academia itself, what about the impact on biology research more broadly? Has AlphaFold 2 genuinely changed how biologists work day to day?
oh, absolutely. Undeniably, it has transformed the landscape. It's an incredibly powerful prediction tool. It gives researchers high-quality structural models, often in minutes or hours, for proteins that might have taken months, years, or even been impossible to solve experimentally before.
But it hasn't put experimental structural biologists out of a job. The source was clear. It's not omniscient, and it actually emphasized the need for them.
That's a really crucial point. AlphaFold 2 is a fantastic predictor, a hypothesis generator. It is not a substitute for experimental validation. Structural biologists haven't become obsolete. They've gained an incredibly powerful new tool. It's maybe like the Human Genome Project. Sequencing the genome didn't make geneticists redundant. It gave them this vast ocean of data to explore and interpret. AlphaFold provides a similar flood of structural data.
So it speeds things up. Allows new questions.
Massively. Having a good structural model readily available lets researchers form hypotheses much faster. How does this protein work? How might this mutation affect it? How does it interact with other molecules? You can design targeted experiments much more efficiently instead of waiting, potentially for years, for an experimental structure. And we're already seeing it used widely. Papers using AlphaFold predictions to study diseases, design new enzymes, understand complex biological systems.
And what about drug discovery? That was touted as a huge potential application area. Structure-based drug design needs those 3D shapes.
Yes, that's a massive area of interest and potential impact. If you know the precise 3D structure of a protein involved in a disease, you can computationally screen or rationally design small molecules, potential drugs that bind specifically to it and modulate its function, maybe inhibit it or activate it.
Like designing a key for a specific lock.
Exactly. Knowing the lock's structure is a huge advantage. And we are seeing early examples. The source mentioned Feng Zhang's lab using AlphaFold 2 predictions to help engineer a novel molecular syringe for drug delivery. The full impact on the drug pipeline will take time to realize, but the potential is certainly enormous.
Okay, so incredible excitement, real acceleration in research. But like any powerful new technology, it's not perfect. It has limitations, right? It's not a magic 8-ball for protein structures.
Yeah, definitely not. And it's really important, as the source cautions, to approach these predictions with caution and humility. Scientists are still actively figuring out its boundaries, where it works well and where it falls short.
So what are some of the key limitations people need to be aware of?
Well, one really critical one, especially relevant given all the excitement about drug discovery, relates to predicting exactly how drug molecules bind to proteins, what's technically called the binding pose.
That's a great question. Let's break it down. If AlphaFold 2 predicts the protein's overall structure, even the binding pocket shape, so accurately, why wouldn't it automatically tell you precisely where a drug fits inside that pocket?
It seems intuitive that it should, but it turns out to be more complicated. A recent study in the journal iLife specifically investigated this. They focused on GPCR's G-protein-coupled receptors, which are a huge class of drug targets. They asked, if we take an AlphaFold 2 predicted structure of a GPCR and then use standard computational docking methods to predict how known drug molecules bind to it, how accurate is that predicted binding pose compared to reality?
And what did they find?
The findings were really interesting and quite sobering. First, the good news. They confirmed that AlphaFold 2 models often predict the shape of the binding pocket itself very accurately. Almost as good as experimental structures, even if those experimental structures were determined with different molecules bound.
Okay, so the pocket looks right in the prediction.
Yes, but here's the crucial part. When they actually tried to dock drug molecules into those accurately predicted pockets, the accuracy of the predicted binding pose, the exact orientation and position of the drug molecule within the pocket was surprisingly poor.
Poorer than expected.
Significantly poorer. It was found to be not statistically better than docking into older, often less accurate, traditional homology models. And critically, it was much less accurate than docking into actual high-resolution experimental structures.
Wow. So even with a highly accurate protein structure prediction from AlphaFold 2, using current docking tools on that static prediction doesn't reliably give you the right drug binding mode.
That's the main takeaway from that study for that specific but very important application. The source concludes that for structure-based drug design, which often needs atomic-level precision to understand interactions, using AlphaFold 2 models currently yields substantially poorer accuracy for predicting binding modes compared to using experimental data.
Why would that be if the pocket shape is right?
It's likely due to a few factors. Proteins aren't perfectly rigid. They can flex and subtly change shape when a drug binds something called induced fit. Predicting that subtle change is hard. Also, accurately sampling all the possible ways a flexible drug molecule could sit in a pocket is computationally challenging. Standard docking methods applied to a single static predicted structure might just not capture these complexities well enough yet.
So for drug developers needing that fine-grained detail, relying solely on docking to an AlphaFold 2 model might be risky. It might give misleading results about how the drug actually binds.
That seems to be the current consensus. If you have a good experimental structure or even a high-quality homology model built by an expert, those might still be preferred for tasks demanding very precise binding pose information.
But AlphaFold models are still useful.
Oh, absolutely. A huge advantage is their availability. The AlphaFold database has predictions for nearly every protein in the human proteome and many other organisms. They're instantly accessible. And if there's no experimental structure or template available, the AlphaFold model might be your only starting point, even with the binding pose limitation. So, incredibly valuable, but with a key caveat for this specific demanding task.
That's a really important nuance. Hey, what about proteins that aren't just single static structures? Many proteins move, change shape, or are even floppy. How does AlphaFold 2 handle those?
That's another major area where it has limitations. Because it was primarily trained on the protein data bank, which mostly contains static snapshots of stable structures, it struggles with proteins that are inherently dynamic or flexible.
Like those weird fold-switching proteins you hear about.
Exactly. These are fascinating proteins that can adopt two or more completely different, stable 3D folds using the exact same amino acid sequence. They might switch shape to bind different partners or perform different functions. The source mentioned one called RAH, which switches between alpha helices and beta sheets. This kind of behavior directly challenges the simple one sequence, one structure idea, and AlphaFold 2, trained on single structures, generally predicts these very poorly.
And then there are the proteins that don't really have any stable structure, the floppy ones.
Right. Intrinsically disordered proteins, or IDPs. Or proteins with long disordered regions. These don't fold into a single, well-defined shape. They exist as a dynamic ensemble of conformations, constantly wiggling and changing the source. Use the analogy of an air dancer outside a car dealership.
Toggles. Okay, I can picture that.
And they're incredibly common. Something like 44% of human proteins have a significantly disordered region. Historically, they were often overlooked because they were hard to study experimentally, maybe a little bit annoying, as the source put it. AlphaFold 2 is actually quite good at predicting which regions of a protein are likely to be disordered, but it doesn't tell you what that ensemble of disordered states looks like.
So it can identify the floppiness, but not describe the flopping.
That's a good way to think about it.
What about small changes, like single amino acid mutations? We know those can cause diseases. How does AlphaFold 2 handle those?
That's another known weakness. It's described as being relatively blind to the effects of single-point mutations. Changing just one amino acid can sometimes have quite dramatic effects on a protein's stability, its structure, or its function. But AlphaFold 2 will often predict the exact same 3D structure for the original protein and the single mutant version. It doesn't reliably capture those subtle yet potentially critical structural consequences.
Okay, so summing up the limitations. Precise drug binding prediction is still a major challenge. It struggles with proteins that change shape or have no shapes at all, and it's not sensitive to the effects of small mutations. It feels like, as the source said, while AlphaFold 2 tackled the core problem of predicting a single static structure, the goalpost has moved to these more complex dynamic aspects of protein behavior.
Exactly. It solved one grand challenge, only to highlight the next set of even more intricate challenges in understanding protein biology.
Which naturally leads us to the next generation, AlphaFold 3.
Yeah.
This was just announced very recently, May 2024.
That's right. And the descriptions are impressive, superior in accuracy and more powerful than its predecessor. The really big news, though, is its dramatically expanded scope.
Expanded scope, how much broader than just single proteins?
AlphaFold 3 aims to go way beyond single proteins. It's designed to predict the structure and the interactions of basically all of life's major molecules working together in complexes.
All of them. Like what?
Proteins, of course, but also DNA, RNA, and importantly, small molecules, things like ions, metabolites, and yes, ligands, which includes drug molecules.
Whoa, okay, so it's not just protein structure, it's protein interactions, protein DNA, protein RNA, and potentially protein drug interactions, all predicted by the same system. That sounds like a massive leap.
It really is. This ability to model how all these different types of molecules come together in biological assemblies is being hailed as potentially transformative for understanding biology and especially for things like drug design and developing new therapeutics, gaining unparalleled insights, as they say, into structural dynamics and interactions.
And it's built on similar AI foundations. Advanced deep learning, maybe using models like Google's Gemini?
Yes, it leverages the latest state-of-the-art AI architectures developed at Google DeepMind. But with these new capabilities, especially trying to model interactions and using newer AI techniques, there are also some new considerations, potentially new risks.
What kind of risks or limitations are being discussed with AlphaFold 3?
One issue that's been raised, possibly related to some of the underlying AI techniques like diffusion models that AF3 might use, is the potential for hallucination.
Hallucination.
Yeah.
In a structure prediction model, what does that mean? Like it makes things up?
Essentially, yes. It means the model might generate structures or interaction modes that look chemically plausible, maybe even energetically favorable on the surface, but they don't actually reflect biological reality. They don't exist or happen in nature.
So it could create convincing fakes.
That's the concern. The source mentioned that even with lots of training data, these kinds of generative models can sometimes produce outputs that look good but are incorrect. It really emphasizes the ongoing, perhaps even increased, need for careful experimental validation of AlphaFold 3's predictions. You can't just assume the output is biologically true.
That makes total sense. The more complex the prediction, the more critical the validation. Now, there's also been quite a bit of discussion, maybe controversy, around the accessibility of AlphaFold 3, right, compared to AlphaFold.
Yes, that's become a major talking point in the scientific community. Unlike AlphaFold 2, where DeepMind released the full source code publicly,
Which was widely praised.
the full code for AlphaFold 3 has not been made publicly available, at least not initially. Access is currently provided primarily through the AlphaFold server, a web-based platform. And crucially, use of this server is restricted to non-commercial research only.
Non-commercial only. So researchers working in pharmaceutical companies or biotech startups trying to discover new drugs, they can't use the AlphaFold server or download the code to run it themselves.
Based on the initial terms, no. They can't use the server for commercial purposes, and the full code isn't available for them to integrate into their own internal drug discovery pipelines. This restricted access, especially the non-commercial clause, has sparked concerns that it could potentially stifle innovation and broader application, particularly in industry where rapid translation is key.
So the argument is that open access, like with AF2, allowed everyone, including commercial entities, to build upon it and innovate faster.
That's the core of the debate. Many researchers feel that allowing unrestricted access, including commercial use, would accelerate progress much faster across the board, leading to quicker development of new medicines and technologies.
It feels like it brings us back to that earlier tension we discussed between proprietary development in industry and the principles of open science in academia. DeepMind got a lot of goodwill for open sourcing AF2. This move with AF3 seems to be shifting the balance back towards keeping the most advanced tool somewhat more controlled.
It definitely raises complex problems. How do you balance the massive investment required to create tools like this within a commercial company against the scientific community's desire for open access to accelerate progress for everyone? There's no easy answer, and it's a very active discussion right now.
Absolutely. Okay, looking beyond AlphaFold 3 even, where does this whole field seem to be heading? What's the next grand challenge or vision?
Well, one really exciting potential future direction is moving towards what some are calling structural systems biology.
Structural systems biology.
Yeah.
What does that entail?
The idea is to go beyond modeling just individual proteins or pairs of molecules and start trying to model the complex web of interactions between many different proteins, DNA, RNA, metabolites, everything, all interacting together within the dynamic context of a living cell.
Wow, so simulating the molecular machinery of the cell, almost like a working ecosystem.
That's the ambition. If we could do that, we could start predicting the consequences of changes like a mutation in one protein or the introduction of a drug on the behavior of entire cellular pathways, or even the whole cell. This could be incredibly powerful for understanding complex diseases like Alzheimer's or Parkinson's, which involve disruptions across many interacting components.
It really feels like these tools aren't just solving old problems. They're enabling us to ask entirely new kinds of questions, questions we couldn't even properly formulate or address before.
Absolutely. The ability to predict structures for potentially millions of uncharacterized proteins in sequence databases could reveal entirely new biological functions, new pathways, new connections we never suspected.
It makes me think of that analogy you mentioned earlier from the source, comparing this to the genomics revolution. We got the genome sequence, this fire hose of data, and then spent decades figuring out what it all meant. Now, maybe we're entering an era of a fire hose of structural and interaction data.
That feels like a very apt comparison. We're potentially generating structural insights much faster than we can fully experimentally validate or functionally interpret them all, which again just highlights how crucial that experimental validation remains. Prediction is powerful, but it needs to be grounded in empirical reality, especially given all those limitations we talked about: dynamics, disorder, mutations, interaction details.
AI is an accelerator, not a replacement for science itself.
Precisely.
We talked about drug discovery and the binding pose issue. Even with that limitation for designing new drugs, Is there still potential for using predicted structures, maybe from AF2 or AF3 for repurposing existing drugs, finding new jobs for old medicines?
Yes, that's definitely an area people are exploring. Even if predicting the exact binding pose is tricky, you can still use computational docking to screen large libraries of already approved drugs against predicted structures of various proteins.
Looking for unexpected matches.
Exactly. Maybe a drug approved for condition X shows some plausible binding to a protein implicated in condition Y. It doesn't prove it will work, but it generates a testable hypothesis much faster than random screening. It could suggest new therapeutic uses or maybe even flag potential off-target side effects. Companies and research groups are definitely using AF models as part of their toolkit for this kind of structure-based repositioning, even if further work is needed to refine the interaction modeling.
So the models provide valuable starting points, even if the downstream modeling of the interaction itself still needs work.
Right. And another really interesting application area that structure prediction unlocks is finding very distant evolutionary relatives, proteins that are related by function and structure, but whose sequences have diverged so much they're hard to connect.
Why would structure be better than sequence for finding those really distant cousins?
It comes down to a fundamental principle the source highlighted. Structure is 3 to 10 times more conserved than sequence over long evolutionary timescales.
3 to 10 times.
Yeah.
Wow.
As proteins evolve, their amino acid sequence can drift quite a bit, accumulating mutations that don't totally break the protein's function. But the overall 3D fold, the essential architecture required for its job, that tends to be preserved much more strongly.
So you could have two proteins whose sequences look almost unrelated if you just compare the letters, but when they fold up, they adopt a very similar 3D shape.
Exactly that. The source mentioned the classic example of RAD52 and red beta proteins involved in DNA repair. Very low sequence similarity, but their structures are strikingly alike, revealing their shared function. Before reliable structure prediction, finding these remote homologies usually required solving both structures experimentally, which was slow and difficult. Now you can predict the structure using something like AlphaFold, and then use powerful structural comparison algorithms to search for similar shapes in databases of known or predicted structures. These structural searches are much more sensitive to distant relationships than sequence searches like BLAST. The plot in the source material showing sequence versus structure similarity really illustrated this lots of proteins cluster with high structural similarity even at very low sequence identity.
So structure prediction basically supercharges our ability to find hidden evolutionary and functional links across the entire tree of life. That's powerful.
It really is. It opens up whole new ways to understand protein families and infer function for uncharacterized genes.
And thinking about real-world complexity, the sources gave some examples where understanding protein structure is just vital, like antibiotic resistance.
Absolutely. Understanding how bacteria evolve resistance often boils down to understanding changes in protein structure. Maybe the target protein mutates so the antibiotic can't bind anymore. Or maybe a transporter protein changes shape to pump the drug out more efficiently. The data shown on E. coli resistance patterns and those specific mutations listed in genes like GIRA and PARS, like S83L, D87N, S80I, conferring resistance to quinolone drugs, figuring out how to combat that requires knowing the 3D structure of those proteins and exactly how those mutations affect drug binding.
And the example of wastewater bacteria having a larger pangenome, more diverse genes, than clinical bacteria.
Right, suggesting a bigger reservoir of potential resistance mechanisms out there in the environment. Understanding the proteins encoded by those extra genes like that biofilm dispersal protein BDCA I mentioned, with its specific active site residues, predicting their structure and function is key to understanding and potentially countering those threats. Structural prediction helps put a 3D face on all that complex genomic data.
It really loops back to the start. Understanding the chase is just fundamental to understanding function, whether it's normal biology or disease or resistance.
It's a foundational piece of the puzzle.
Okay, so what a journey. We've gone from this decades-old, seemingly intractable protein folding problem through the slow, steady progress and key breakthroughs like coevolution, right up to the seismic shift caused by AlphaFold 2, driven by incredible engineering and resources.
A real paradigm shift.
We've looked at its amazing impact on speeding up research, but also dug into those really important limitations, especially around predicting precise drug binding, handling protein dynamics and disorder, and sensitivity to mutations.
Critical caveats to keep in mind.
And then we saw how AlphaFold 3 is pushing the boundary even further, aiming to model the interactions of basically all of life's molecules, while simultaneously raising new questions about open access versus proprietary tools and the ever-present need for validation.
It's a field moving at an absolutely breakneck pace, building on decades of prior work, both experimental and computational. The predictive tools are astonishingly powerful now, but they achieve their maximum impact when they're used smartly, hand in hand with rigorous experiments.
Absolutely. So here's a final thought, maybe something for you, our listeners, to chew on. We have these incredibly powerful AI tools like AlphaFold, capable of predicting structures and interactions with remarkable accuracy in many cases. But we also know there are limitations dynamics, subtle changes, the need for validation. So how should the scientific community best harness this power? Should the primary focus now shift massively towards experimental validation of AI predictions? Or are there still fundamental gaps in our theoretical understanding, maybe needing non-AI approaches to truly grasp the deepest principles of that folding code mechanism Anfinson talked about? And maybe most importantly, how do we ensure this incredible progress continues, fostering innovation across both academia and industry to tackle the biggest challenges in biology and medicine?
Those are definitely the big questions shaping the future of this entire field. Lots to think about.
Indeed.
Yeah.
Thank you for joining again.